You know, that feeling when a cold hits you, it's usually this really slow creep.
Right, the gradual onset.
Yeah, exactly. You might feel a little tickle in your throat on a Tuesday, maybe you're a bit sniffly on Wednesday, and by Thursday you're just, you're tired and annoyed.
But you push through it.
You do. You go to work, you apologize to everyone for sneezing, but you function.
And that right there is the classic rhinovirus experience. I mean, it's a nuisance, but it's manageable.
Right. But then there is that other feeling.
Oh, yeah.
The one where you are walking down the street at like 9:00 a.m. feeling perfectly fine, maybe thinking about what you want for lunch, and by noon you are flat on your back.
Completely immobilized.
You are shivering so hard, your teeth chatter, your muscles feel like they've been beaten with a bat, and you literally feel like you've been hit by a truck.
That hit by a truck sensation is a very specific biological signature. That is the hallmark of the influenza virus.
And that's exactly what we are unpacking today for you, because I think there's a lot of confusion out there. We aren't talking about the stomach flu, and we're not talking about the common cold. We are taking a deep dive into the Orthomyxoviridae family. And when I looked at the sheer scale of this thing in the source material you sent over, it honestly blew my mind.
The numbers are staggering.
They really are. The very first line of the text says respiratory illnesses cause more than half of all acute illnesses in the U.S. every single year. Half.
It is a massive statistic. I think we tend to focus our anxiety on the scary exotic diseases that make the evening news, Ebola, Zika, things like that.
Yeah.
But in terms of raw morbidity, which is just the medical way of saying how many people actually get sick and mortality, the orthomyxoviruses are the undisputed heavy hitters.
They really are.
They are this constant, invisible enemy that we have we've made a truce with them. Largely because we underestimate them.
A truce is a really good way to put it. We treat it like it's just inevitable weather. Oh, it's flu season. But the mission of this deep dive is to completely shatter that complacency. We really want to understand why this specific virus is described in the medical literature as a major determinant of global morbidity and mortality. It's not just bad luck that you get sick. It's a machine.
It is an incredibly highly evolved machine, a shape-shifting, genetic splicing, identity-stealing machine.
Wow.
And to understand why it remains a formidable challenge, despite all our modern medicine, as the text puts it, we have to look under the hood. We need to look at its armor, its engine, and its weaponry.
Okay, before we shrink down to the molecular level, though, we need to clear up the lineup. Because I think most people hear the flu and think it's just one monolithic thing.
Right, they think it's a single virus.
But there are actually 3 distinct characters in this story, right? Types A, B, and C.
Correct. And they are definitely not created equal. They are categorized based on their internal immunologic proteins, but their behavior out in the real world is vastly different.
So introduce us to the family. Who is the annoying little brother and who is the serial killer?
Well, the annoying little brother would definitely be influenza C.
Okay.
It's what we call antigenically stable. That means it doesn't really change its face much. It causes minor illness, a few sniffles, maybe a light cough.
So nothing crazy?
No, it rarely causes epidemics, and it's not really a major public health headache at all. It's just background noise.
Okay, so we don't really worry about C. What about type B?
Influenza B is sort of the middle child. It is strictly a human problem. It only infects us.
Only humans.
Yes. It does change a bit over time, which we call antigenic drift, but it's generally much more stable than the big boss. You can cause seasonal epidemics. It can absolutely make you very sick, but it usually doesn't have the capacity to cause those global civilization-altering pandemics.
Which leaves us with type A.
The monster, Influenza A. This is the one that keeps virologists and public health officials awake at night.
Why?
First, it is antigenically highly variable. It changes its disguise constantly. But the scariest thing about type A isn't just that it changes, it's where it lives.
The reservoir.
Exactly. Type A doesn't just infect humans. It infects ducks, chickens, pigs, horses, seals, whales. It has a massive animal kingdom playground.
Seals and whales. I had no idea.
Oh, yeah. And as we will discuss later, that ability to jump between species is exactly what gives it the potential to be catastrophic.
Okay, so type A is our primary target today. Now let's get visual for the listener. If I had a super-powered electron microscope and I zoomed in on an influenza A particle, what am I actually looking at?
You're looking at something incredibly tiny, about 100 nanometers in diameter.
Yeah.
But the very first thing you'd notice is that it doesn't have a rigid geometric shape like a lot of other viruses do.
Like those ones that look like little lunar landers.
Exactly, the bacteriophages. No, influenza is pleomorphic.
Pleomorphic, meaning many shapes. Right?
Yes.
Yeah.
Usually it looks roughly spherical, like a little ball, but sometimes it stretches out into long filaments or it just looks totally irregular and shape-shifting. It's highly adaptable. And this ball is wrapped in a coat of lipid envelope.
And lipid means fat.
Essentially, yes. It's a greasy outer membrane. And here is a truly fascinating detail from the source. The virus doesn't actually make this envelope itself.
It doesn't.
No, it steals it. When the virus finally bursts out of your infected cell, it wraps itself in a piece of your own cell's plasma membrane.
So it's wearing a jacket made of me.
It is wearing a jacket made of you.
That is incredibly creepy.
It is. But it's not just a smooth ball of stolen fat, right? It's covered in stuff.
Spikes.
Right.
Yes.
It is heavily studded with spikes, hundreds of them sticking out.
Yeah.
From the outside, it looks a bit like a microscopic naval mine.
Okay, I can picture that.
Those spikes are the weapons, and we will definitely get into how those work in a moment. But first, we really have to look inside. We have to look at the guts of the virus.
Yes, this is the part of the research that honestly confused me at first. Most viruses I've read about, their genetic code is like a scroll. It's one long, continuous string of DNA or RNA. You unroll it, you read it from start to finish, and you're done. The flu is different.
Flu is fundamentally different. It has what we call a segmented genome.
Meaning it's broken up into pieces.
Right. Influenza A and B contain 8 separate segments of single-stranded RNA. Type C has 7, but again, let's focus on A and its 8 segments.
So instead of a single scroll, it's more like, what, a deck of cards? Or chapters in a book?
A deck of cards is a fantastic analogy. Imagine playing with a deck that only has 8 cards, but every single card is absolutely vital. You need the whole hand to play the game.
And why is that specific structure so important? Does it actually help the virus?
It is the single most important evolutionary feature the virus has. Think about it. If you have a single long scroll of genetic code, it's really hard to swap out just the middle paragraph without messing up the whole text.
Sure.
But if you have a deck of cards, you can shuffle. If 2 entirely different flu viruses infect the exact same cell at the same time, they can both drop their cards on the table and mix them up.
Oh, wow.
We call this process genetic reassortment, and it is the exact mechanism that creates pandemics.
We are definitely going to circle back to that shuffle because the implications of that are terrifying. But I want to stick on the genetics for just a second more. The text called this RNA negative sense. What does that actually mean?
In molecular biology, positive sense RNA is like a book written in plain English. If a human cell sees it, the cell's machinery can just read it immediately and start manufacturing proteins based on the instructions. It just makes sense to the cell.
Okay, so positive sense is ready to go.
Exactly. Negative sense, on the other hand, is like a book written entirely in mirror image text. If your cell looks at it, basically says, I can't read this, this is gibberish. It is the template, but it's not the readable message.
So if the host cell can't read it, how does the virus even work? How does it replicate?
Well, the virus has to come prepared. It knows it can't just rely on the host cell right out of the gate. So packed inside that virus particle, physically attached to those 8 RNA segments, is a specialized enzyme.
The text called it an RNA-dependent RNA polymerase.
Yes. Think of it as a decoder ring or a translator. The virus brings its own molecular photocopier that takes the mirror image text, the negative sense, and transcribes it into positive sense, which is the readable message, so the host cell can finally understand it.
That makes total sense. So it literally packs its own translation tools.
Exactly. These polymerase proteins, they are specifically named PB1, PB2, and PA in the text. They are attached right to the ends of the RNA strands. They are locked and loaded, ready to go the absolute moment the virus enters the cell. Without them, the viral RNA is just inert. It would do nothing.
That is such a cool, specific detail. It's not just a bag of random genes. It's a bag of genes plus the exact tools needed to read them.
Precisely.
Okay, so that's the inside. The segmented negative sense deck of cards with its own translators. Now let's talk about the outside. The spikes on the lipid envelope. You called them the weapons earlier.
The keys to the kingdom, really. This is where the rubber meets the road.
Yeah.
Or more accurately, where the virus physically meets your lung tissue.
Right.
There are 2 main types of spikes protruding from that lipid envelope, and their names are actually the basis for how we name flu strains in the news. They are hemagglutinin and neuraminidase.
HA and NA. So when I hear about H1N1 or H5N1, this is exactly what we're talking about.
Precisely. Let's start with hemagglutinin, the HA spike. This is the most abundant spike on the surface. It makes up about 25% of the total viral protein.
And what does it look like structurally?
It's a rod-shaped trimer. That means it is composed of 3 identical protein chains tightly twisted together, almost like a braided rope.
And what is its primary job?
It is the grappling hook. Its only real job at the beginning is to float around, find a specific target receptor on the surface of your cell, and latch on tight.
What exactly is it looking for?
It's looking for a molecule called sialic acid.
Sialic acid. Is that something rare in the human body?
Not at all, and that is exactly the problem. Sialic acid is a tiny sugar molecule that sits on the very end of glycoprotein chains on the surface of almost all of our respiratory cells.
Oh, so it's everywhere.
It's everywhere. But, and here is a very crucial nuance we have to understand, the physical shape of that sialic acid receptor varies between different animal species.
How so?
Well, humans have one specific structural type of sialic acid receptor in our upper airways. Birds have a slightly different structural type in their intestinal tracts. The HA spike of a given virus is highly specialized. It is shaped to fit only one specific type of sialic acid, very much like a specific key fitting into a specific lock.
So the HA spike bumps into a human respiratory cell, feels that specific human-type sialic acid, and locks on.
Right.
But just latching on to the outside of the cell isn't enough, right? The virus has to get its deck of cards inside.
Correct. And this is where the HA protein transforms from a simple grappling hook into something much more aggressive, like a harpoon. The source text describes a specific fusion peptide that is physically buried deep inside the structure of the HA molecule.
Buried, so it's hidden away at first.
It is totally hidden during the initial approach. It's a hydrophobic or water-repelling sequence of amino acids. But once the virus attaches to the cell surface, the cell actually swallows the virus in a little bubble called an endosome.
Just pulls it inside.
Yes. And once inside, the cellular environment changes. The cell naturally pumps acid into that endosome to try and digest whatever it just swallowed. The pH drops significantly.
The outline called this the acid test.
That's a great name for it, because this severe drop in pH is the mechanical trigger. The acidic environment causes the entire HA molecule to undergo a massive, irreversible physical shape change. It literally snaps open like a switchblade.
Whoa.
It exposes that previously hidden fusion peptide, which then violently plunges into the membrane of the host cell's endosome and pulls the viral envelope and the cellular membrane tightly together until they completely fuse into one continuous sheet.
That is incredibly violent imagery for a microscopic molecule. It acts exactly like a spring-loaded trap.
It really is molecular engineering at its absolute finest. The source text explicitly mentions that the HA protein contains exactly 566 amino acids.
566.
Yes, and every single one of those amino acids is positioned there through millions of years of evolution to ensure this mechanical switchblade flip happens exactly when it's supposed to and only under those specific acidic conditions.
That's amazing. Okay, so the HA grappling hook gets us attached, and the fusion peptide gets us inside. What about the other spike, the NA neuraminidase?
If HA is the grappling hook and harpoon, NA is the machete, or maybe the scissors. Structurally, it is a mushroom-shaped tetramer.
Tetramer meaning 4 parts.
Exactly, 4 parts acting together on a stalk. And fundamentally, it is an enzyme, a sialidase.
Meaning it cuts sialic acid.
It cuts sialic acid.
Wait, I'm genuinely confused here. The HA spike just went through all that trouble to grab onto sialic acid. Why on earth would the virus pack an enzyme specifically designed to cut and destroy the very thing it uses to hold on?
It all comes down to timing. HA is purely for the beginning of the infection, getting into the cell. NA is for the end of the life cycle.
Okay, walk me through that.
Imagine the virus has successfully replicated inside the host cell. It has hijacked the machinery and made tens of thousands of copies of its genetic deck of cards and all its proteins. These new baby viruses push out of the cell membrane, a process called budding.
Right.
But as they bud out, their own surfaces are completely covered in brand new HA spikes.
Oh, so as soon as they exit, they would just immediately grab onto the sialic acid on the dying cell they just left.
Exactly. Without intervention, they would instantly stick to the dying cell's surface. Or worse, they would stick to the sialic acid receptors on each other, forming a massive, useless clump of trapped viruses. They would never be able to spread to the next cell or to the next person.
Enter the scissors.
Enter neuraminidase. The NA enzyme comes in, and snips the sialic acid tether. It chemically cuts the receptor right off the host cell surface, physically releasing the newly formed virus to float away and find a fresh, uninfected victim. It basically burns the bridge behind it so the progeny don't get stuck.
That is absolutely diabolical. HA to enter, NA to escape.
It is a perfect binary system. There is also an interesting theory mentioned in the text that NA helps the virus initially penetrate the thick mucus layer in our respiratory tract.
How so?
Well, respiratory mucus is absolutely loaded with free-floating sialic acid molecules. It acts as a decoy defense mechanism. The HA spikes grab the mucus and get stuck. But NA might act like a molecular snowplow, constantly cutting those decoy tethers, allowing the virus to slice through the mucin layer to finally reach the actual vulnerable epithelial cells underneath.
Wow. Okay, so we have the grappler and the cutter. But there are a couple of other structural proteins mentioned in the anatomy section of the text. The M proteins, M1 and M2.
Right, the matrix proteins. M1 is the major matrix protein. It forms a rigid protein shell right underneath that stolen lipid envelope.
So it's like the skeleton.
Exactly. It provides the physical structure. It's the internal scaffolding that keeps the whole spherical particle from just collapsing or falling apart in the environment.
And what about M2?
M2 is a tiny but absolutely critical protein. It forms an ion channel. Basically, it's a pore or a microscopic tunnel that goes right through the lipid envelope.
Why does a virus need a tunnel through its own armor?
Remember that acid test we just talked about? We said the host cell's acidic environment inside the endosome causes the HA spike on the outside to flip.
Right.
But that acid also needs to get inside the virus particle itself. It needs to reach the internal core to loosen up the M1 scaffolding and free up those 8 RNA segments so they can be released into the cell.
Ah, so the M2 channel is the tunnel that lets the acid inside.
It is the tunnel. It specifically allows protons, hydrogen ions to flow from the acidic endosome outside the virus directly into the interior of the virus particle. It acidifies the viral core.
And if that doesn't happen...
Without a functioning M2 channel, the virus stays completely locked up. It manages to enter the cell, but it can never uncoat its genetic material. It just becomes a harmless dud.
Which I imagine makes that M2 tunnel a pretty tempting target for pharmaceutical drugs.
It was a huge target. We will definitely get to how we tried to exploit that later.
Awesome. Okay, so we have essentially built the influenza machine from the ground up. We have the segmented genome, that 8-card deck, the built-in photocopiers, the HA grappling hook, the NA escape scissors, the M1 scaffolding, and the M2 acid tunnel.
That's the complete anatomical picture, yes.
So now let's talk about why this matters to the average person. Why do I need to go to the pharmacy and get a new flu shot every single year? I got the measles shot when I was a kid, and I'm supposedly good for life. Why is influenza such a constantly moving target?
It all comes down to 2 evolutionary concepts that sound similar by name, but are entirely different in terms of their global consequences. Antigenic drift and antigenic shift.
Drift and shift. It honestly sounds like a Fast and Furious movie title.
It does, but it very accurately describes how the virus maneuvers through the human population. Let's start with the slow burn. Antigenic drift.
How does drift work?
It is basically driven by sloppiness. Remember that viral polymerase? The photocopier enzyme that reads the negative sense RNA.
The one that translates the mirror image, yes.
Right. Well, it is an incredibly fast machine, but it completely lacks a delete key. It lacks any proofreading ability. When human cells replicate our DNA, we have complex enzyme systems to check for errors and fix them on the fly. The flu virus doesn't have that luxury.
So it makes mistakes.
It makes constant mistakes. Every single time it copies its genome, it introduces tiny typos into the genetic code. We call these point mutations.
Just single letters in the code getting swapped out.
Exactly. And importantly, most of the typos that actually matter to us happen in the genes that code for the surface spikes, the HA and NA proteins. Over the course of a flu season, as the virus jumps from person to person, these tiny typos accumulate.
Okay.
So the physical shape of the HA spike on the virus that infects you in December is actually slightly physically different from the one that was circulating back in September.
So the antibodies my body made.
Your antibodies from last year's flu shot or from the infection you had 3 years ago, they circulate in your blood, they squint at this new winter virus, and they say, you look familiar, but something is off.
Like it's wearing a disguise.
The virus is essentially put on a fake mustache. Your immune system might recognize it just a little bit, so maybe it fights it off enough that you don't end up in the hospital, but you still get sick.
That makes total sense.
This relentless gradual drift is exactly what causes the annual seasonal epidemics. It's the entire reason the World Health Organization has to constantly monitor global strains and update the vaccine recipe every single year to try and match the current disguise.
Okay, so drift is the reason for the annual sniffles and the yearly shot. But then there is the nightmare scenario the outline highlights, antigenic shift.
Shift is the absolute catastrophe. Shift is the biological mechanism behind global pandemics. And this brings us right back to the deck of cards.
The segmented genome.
Yes, antigenic shift happens through that process of genetic reassortment. And to truly visualize this, you have to understand the concept of the mixing vessel.
Which, in the source text, is usually a pig.
Often a pig, yes. Let's explain why pigs are the usual culprits. Pigs are biologically unique in this specific context because the respiratory epithelial cells in their windpipes have receptors for both avian bird flu viruses and human flu viruses.
Oh, because earlier we said humans have one receptor type and birds have another.
Right. Humans have the alpha-2 sex receptors, birds have the alpha-2/3, pigs have both sitting right next to each other. They act as a universal biological adapter.
Okay, so imagine a scenario on a farm. You have a pig in a pen, a wild duck flies overhead, droppings fall into the pig's trough, and the pig inhales it. The pig gets infected with an avian flu, a type A bird virus.
Perfect setup.
But at the exact same time, the human farmer, who happens to have the seasonal human flu, sneezes on that exact same pig. The pig gets the human flu.
Now, zoom way in. Inside one single respiratory cell in that pig's airway, both of those entirely different viruses are replicating at the exact same time. They're both unpacking their genetic decks of cards in the same space.
So you have 8 RNA segments from the bird virus and 8 RNA segments from the human virus just floating around together in the same cellular cytoplasm.
Yes. And when that host cell starts frantically assembling the new progeny viruses, it doesn't know which parts belong to which virus. It just grabs 8 segments at random to fill the viral envelope.
It just shuffles the deck.
It shuffles the decks together. So it might grab, say, 7 internal RNA segments from the human virus, meaning this new virus knows perfectly well how to replicate and spread efficiently in mammalian cells. But for that crucial 8th segment, it grabs the HA spike gene from the bird virus.
So you end up with a mutant virus that spreads easily from human to human, but it's wearing an exterior spike coat that no human immune system has ever encountered before.
Exactly. It's not just a fake mustache anymore. It is a complete face transplant. The human population has absolutely zero pre-existing immunity to this novel avian HA spike.
The antibodies from last year's flu shot.
Totally useless. They don't recognize this bird spike at all. They just flow right past it.
And because absolutely no one on Earth is immune to it, it spreads like absolute wildfire.
That is a pandemic. That massive antigenic shift is exactly what happened in 1957 with the H2N2 pandemic, in 1968 with the H3N2, and most recently in 2009 with the H1N1 swine flu.
The 2009 one was particularly crazy, wasn't it? The source mentions it was a quadruple reassortment.
It was incredibly complex. It contained genetic segments derived from North American swine flu, Eurasian swine flu, avian flu, and human flu, all mashed together. A true Frankenstein virus.
And the source material is very specific here. Only type A influenza can do this.
Correct. Type B and type C generally do not infect animals in the same way, so they simply don't have access to this vast global library of spare animal parts to swap with. Type A has the entire animal kingdom to borrow lethal traits from.
That is terrifyingly efficient. Now, I want to briefly touch on the code-breaking aspect. You see these ridiculously long names in the news or on vaccine vials. A Hong Kong 0368 H3N2, it looks like a serial number on a car part.
It is essentially a full intelligence dossier. It tells virologists everything they need to know about that specific virus at a glance.
Break it down for me.
Sure. The A tells you the immunologic type, so we immediately know it's the dangerous one capable of shift. Hong Kong is a geographic location where that exact strain was first isolated from a patient.
Makes sense.
03 is simply the laboratory strain number from that location. 68 is the year it was isolated, 1968 in this case. And finally, the part in parentheses H3N2 tells you exactly which surface weapons it's equipped with. Hemagglutinin type 3 and neuraminidase type 2.
So if a scientist reads that string of text, they instantly know the entire origin story and armament of the villain.
Exactly. It's standard nomenclature worldwide.
Okay, let's move into the action movie portion of the script. Section 3 in our outline. The invasion, the replication cycle. We've talked about all the individual parts, but how does the heist actually go down inside the human body?
It is incredibly fast. The source material points out that an entirely new generation of progeny viruses is produced within just 8 to 10 hours of the initial entry. That is a lightning-fast biological turnaround.
Wow. Okay, so step 1 is attachment, which we covered, the HA spike grabbing the sialic acid receptor.
Right. And then the cell, either thinking it's bringing in something useful like a nutrient, or just being entirely tricked by the surface chemistry, engulfs the virus. This is called receptor-mediated endocytosis. The virus is now inside a tiny bubble, the endosome, cruising deeper into the host cell.
Then comes step 2, the acid test we talked about.
Yes, the cell naturally acidifies that endosome. The virus uses that exact offensive acid against the cell. The acid flows through the M2 channel, the internal core loosens, the HA spring-loaded mechanism flips, the membranes violently fuse, and the 8 viral RNA segments are dumped straight into the cell's cytoplasm.
Now, usually when I read about RNA viruses, they stay in the cytoplasm, it's safer there, and all the protein-making factories are out there. But flu is arrogant. It doesn't stay there.
No, it goes straight to the host cell's command center. This is a highly unique feature of orthomyxoviruses. The viral RNA segments physically travel into the nucleus of the host cell.
And once it gets into the nucleus, it commits a molecular crime that I found honestly shocking. The text calls it cap snatching.
It really is the ultimate form of identity theft. Here is the fundamental problem the virus faces. Its viral polymerase, that photocopier enzyme, cannot just start writing new messenger RNA completely on its own from scratch.
Why not?
It mechanically requires a primer, a tiny specific starter sequence of code to physically attach to before the engine can start running. But the virus didn't pack one.
So where does it get the primer?
It steals it from the host. It looks around the nucleus at the host cell's own freshly made messenger RNA. These are the vital instructions the host cell is desperately trying to send out to the cytoplasm to keep itself alive and functioning.
Okay.
The viral polymerase enzyme physically grabs onto one of those host messages and literally cuts off the first few letters of the code. It snips off the 5' cap.
It decapitates the human cell's messages.
It decapitates them. It steals that host cap sequence and chemically pastes it right onto the front of its own viral RNA.
So it's basically using the cell's own login credentials to trick the ribosomes later on into making viral proteins instead of human proteins.
Exactly. And it achieves 2 massive victories with this one single biochemical move. First, it successfully jumpstarts its own massive replication. And second, by systematically decapitating all the host's messenger RNA, it rapidly shuts down the cell's ability to maintain its own basic functions or to send out distress signals to the immune system. It is sabotage and theft perfectly combined.
That is absolutely brutal.
There's a tiny, fascinating detail in the source that really highlights how precise this cap-snatching mechanism is. It specifically mentions that the first 12 to 13 nucleotides at the very ends of all the viral RNA segments are conserved.
Conserved meaning they never mutate, they never change.
Exactly. Even across completely different strains over decades, those specific 12 to 13 letters are functionally identical. They are the exact docking station required for this whole complex cap-snatching machine to physically align. If those 12 letters mutated, the whole system would break and the virus would die.
So it's an Achilles heel, technically.
It is, but it's structurally very well protected by the polymerase complex.
So the heist is successful. The human cell is now basically a zombie factory, frantically churning out viral parts. The newly minted HA and NA proteins are shipped out to the cell surface to create a staging area. The newly copied RNA segments come out of the nucleus. They all meet at the outer membrane.
And then comes step 4, assembly and budding. The internal proteins align. The virus physically pushes out, wrapping itself tightly in that patch of altered cell membrane.
The stolen jacket.
Right. And finally, the NA scissors cut the sialic acid cord, and the brand new virus particle floats free,
leaving behind a dying, completely ruined host cell.
And moving on to infect the neighboring cell. And the cycle repeats exponentially.
Which brings us perfectly to the actual human cost. Section 4 in our outline, the toll. What is actually physically happening inside my body while this microscopic war is raging in my cells?
The virus primarily targets the respiratory epithelium. This is the delicate tissue lining your entire airway, from your nose down into your lungs. But specifically, it really loves to attack the ciliated columnar epithelial cells.
The ones with the tiny little hairs on them.
Yes, the cilia. You have to think of those cilia like a microscopic lawn of grass that is constantly waving and beating upwards. Their entire job is to sweep mucus, trap dust, and incoming pathogens up and out of your deep lungs so you can swallow them or cough them out. They are your primary janitorial staff.
And the flu virus.
The flu virus basically mows the lawn. It infects and completely kills those ciliated cells. It strips the respiratory epithelium bare down to the basement membrane.
So suddenly I have absolutely no mechanism to clear the mucus out of my lungs.
Exactly. That's exactly why you develop that deep, painful, hacking cough. Your body is desperately trying to manually clear the airway because the automatic sweeping system has been destroyed. And the source material explicitly notes that even after your immune system clears the virus entirely, it takes about a full month for that delicate ciliated tissue to actually repair and regenerate itself.
Wow, a whole month?
Yeah, that's exactly why a post-flu cough can linger for weeks and weeks. Your windpipe is literally raw tissue.
Now, we need to clearly distinguish between a bad cold and the actual flu again, because the clinical symptoms really tell the story of this cellular destruction.
The absolute key word for influenza is abrupt. A cold creeps in over days. The flu kicks the front door down. The incubation period is remarkably short, just 1 to 4 days after exposure, and then boom, sudden high fever, severe headache, a dry, non-productive cough, and the defining feature, severe myalgias.
Muscle aches?
Terrible muscle aches. And here's the truly interesting physiological thing about that. The influenza virus itself isn't usually replicating inside your muscle tissue. The viral infection is physically localized in your respiratory tract.
So why do my legs and back hurt so much I can't stand up?
Cytokines.
Cytokines, what are those?
They are powerful chemical messenger proteins.
Okay.
When your immune system finally realizes the scale of the viral invasion happening in your lungs, it panics. It dumps massive amounts of these inflammatory cytokines directly into your bloodstream to mobilize a system-wide defense. The high fever, the shivering chills, the severe muscle aches, that is all friendly fire. It's your own body going to war, raising the temperature, and causing systemic inflammation to try and stop the virus before it spreads further. It's a scorched earth defense tactic.
It makes you feel like you're dying to save your life.
Essentially, yes.
Let's talk about the severe complications. Because historically, the flu virus itself isn't always the direct executioner. Sometimes it just kicks the door open for the actual executioner to walk in.
That is a very grim but accurate way to put it. The leading cause of death associated with influenza infections is pneumonia. And there are 2 distinct ways this happens. One is primary viral pneumonia.
Which is when the flu virus itself just keeps moving down and eating the deep lung tissue?
Yes. The viral infection spreads unchecked down into the lowest alveoli, destroying the thin tissue where oxygen exchange actually happens. The lungs fill with fluid and immune cells. This is incredibly severe and often rapidly fatal. But luckily, it is less common.
So what's the more common killer?
The much more common killer is secondary bacterial pneumonia.
The classic 1-2 punch.
Exactly. Remember how we just said the flu virus mows the lawn and violently strips away the protective cilia, and how it leaves behind a massive layer of dead cellular debris and nutrient-rich fluid sitting stagnant in the lungs?
It sounds like it's creating a giant petri dish inside your chest.
It creates the absolute perfect undefended buffet for bacteria. Bacteria that are normally just hanging around completely harmlessly in your nose, or throat-specific ones highlighted in the text like Staphylococcus aureus, Streptococcus pneumonia, or Haemophilus influenza. They see this massive opening.
When defenses are down.
The mechanical defenses are completely gone. So those bacteria easily travel down the airway into the deep lungs where they normally could never survive, and they aggressively set up shop and multiply in all that dead tissue.
So a patient might actually survive the initial viral attack, start to feel a tiny bit better, and then suddenly crash and die from a massive bacterial invasion a week later.
That biphasic pattern sick, briefly better, then critically ill, was exactly the pattern for millions of victims in the 1918 pandemic. They survived the viral phase in the first week, but then rapidly succumbed to secondary bacterial pneumonia because antibiotics hadn't been invented yet.
Man, there's another specific complication mentioned in this section that really scares me because it directly involves children. Reye syndrome.
Yes, this is a massive public health lesson that everyone needs to know. Reye syndrome is a rare but absolutely catastrophic condition. It involves acute encephalopathy, which is massive, rapid swelling of the brain, combined with severe fatty degeneration of the liver. The mortality rate is terrifyingly high, up to 10 to 40%.
And what actually causes it? Is it the flu virus mutating in the brain?
No, it's not the virus directly. Decades ago, epidemiologists found a very strong, undeniable link between the development of Reye syndrome and the use of salicylates, specifically aspirin to treat viral fevers in young children and teenagers.
So, parents treating the flu symptoms with the wrong over-the-counter drug triggered a lethal metabolic reaction.
Yes. The combination of the influenza virus, or sometimes the chickenpox virus, and aspirin in a developing child's system caused this catastrophic systemic failure.
That's horrifying.
It is. Once the medical community realized this and started aggressively warning parents to absolutely never give aspirin to children with flu-like symptoms, the cases of Ray's syndrome almost completely vanished overnight.
So the golden rule stands.
The rule is absolute. If a child or teenager has a viral fever, you use acetaminophen like Tylenol or ibuprofen. Never, ever use aspirin.
That is a crucial life-saving tip. Let's shift gears slightly to immunity. Let's say I catch the flu, I survive the aches and the potential pneumonia, and I recover. Am I completely bulletproof now?
You are bulletproof against that one specific bullet. Influenza immunity is strictly subtype specific. If you catch an H1N1 strain this winter, your immune system will generate fantastic, long-lasting antibodies against that exact H1N1 configuration. But if an H3N2 strain comes circulating through your town next month, those H1N1 antibodies will basically just wave at it as it goes by. They offer very little cross-protection.
And the source text points out that antibodies specifically against the HA spike are the most important ones to have. Why is that?
Because the anti-HA antibodies are what we call neutralizing. Think back to the mechanics. Because the HA spike is the grappling hook the virus uses to attach to your cells in the first place, if your immune system can flood the area with antibodies that physically stick to and cover up those HA spikes, the virus literally cannot attach.
It's like putting tennis balls on the ends of a walker.
Exactly. It just harmlessly bounces off your cells. You don't even get mildly infected.
What about antibodies against the NA spike the scissors? Do those help?
They are definitely helpful, but they don't prevent the initial infection. They just stop the release. So if you have high levels of anti-NA antibodies, the virus will still successfully infect your cells, but when the newly formed baby viruses try to bud out, the anti-NA antibodies gum up the scissors.
So they can't cut the tether.
Right? The viruses stay trapped on the dying cell, so it severely limits the spread of the virus within your lungs. You will probably still get sick, but you get a much milder case. It's a mechanism of damage control, not total prevention.
Okay, that brings us to Section 5, Diagnosis and Epidemiology. How did the lab detectives actually know exactly which strain of the virus we have circulating?
Well, historically, and actually still today, for massive vaccine production, we relied heavily on embryonated chicken eggs.
Wait, like fertilized eggs?
Yes. A lab worker would physically inject the patient's swab sample deep into a growing chicken egg, put it in an incubator, and wait several days to see if the virus multiplied in the embryonic fluid. It's the gold standard for pure virus isolation.
But that sounds incredibly slow for diagnosing a sick patient today.
It is way too slow for clinical care. Today, the modern gold standard for diagnosis is PCR polymerase chain reaction.
The same tech we use for practically everything now.
Exactly. PCR directly searches for the specific viral RNA genetic code in your nasal swab. It's an incredibly fast, vastly more sensitive than old rapid tests, and a multiplex PCR panel can tell the doctor exactly which type and subtype H1N1 versus H3N2 you have within a couple of hours.
There is one older, really clever lab test mentioned in this section that I want to highlight. The hemagglutination inhibition test, or the HI test.
Yes. So the HI test is a classic piece of virology. It perfectly exploits a strange biological quirk of that HA grappling hook spike.
What's the quirk?
It turns out that the HA spike doesn't just bind to respiratory cells. It will also aggressively bind to the sialic acid found on the surface of standard red blood cells. So if a scientist takes a test tube of liquid red blood cells and drops in live flu virus, the HA spikes will cross-link all the blood cells together. They clump up and form this visible, solid lattice structure that you can see with the naked eye. That process is called hemagglutination.
So how does that become a test for immunity?
Here is the genius part.
Yeah.
If a patient actually has neutralizing antibodies against that specific strain in their blood serum, and you mix the patient's serum with the virus first.
The antibodies cover up the HA spikes.
Exactly. They coat the virus completely. Then, when you pour that mixture into the red blood cells, no clumping happens. The blood stays completely liquid. The patient's antibodies successfully inhibited the hemagglutination.
So a liquid test tube means you're immune, and a clump test tube means you have no antibodies.
Correct. It is a brilliant, visual, low-tech proof of specific immunity.
That is so elegant. All right, let's talk pure numbers. epidemiology. What is the actual global footprint of this virus?
Well, structurally, it's a deeply seasonal disease. In the Northern Hemisphere, it predictably peaks between January and April. In the Southern Hemisphere, it's May through August. Basically, wherever the air is cold and dry, which keeps the lipid envelope stable in the air longer, and wherever people are crowded closely together indoors, the virus thrives.
And what is the annual body count?
It is basically a quiet, continuous massacre. In a completely normal, non-pandemic year, there are 3 to 5 million severe illness cases globally, and between 250,000 and 500,000 confirmed deaths.
500,000 people, every single year.
Every single year. And the economic cost in the U.S. alone is estimated at $10 to $60 million per million population in lost productivity and healthcare costs.
But again, that's just a normal year. We can't possibly do a full deep dive on influenza without talking about the abnormal years, the pandemics. The source outline lists the major recent ones, 1918, 1957, 1968, 2009. But 1918, the infamous Spanish flu, it really stands completely alone in history, doesn't it?
It stands alone as one of the deadliest biological events in human history. It conservatively killed more people than all the combat deaths of World War I combined.
Wow.
Global mortality estimates range wildly from 20 to 50 million, and some modern epidemiologists think it may have been closer to 100 million dead.
And the truly terrifying thing about the 1918 virus wasn't just how many people it killed, it was who it killed. Usually, seasonal flu heavily targets the vulnerable extremes, the very old and the very young.
Right, what epidemiologists call a U-shaped mortality curve on a graph.
Yeah.
High at the extreme ends of age, very low in the middle. But the 1918 pandemic was a W-shaped curve. It caused absolutely massive mortality right in the middle among completely healthy young adults in their 20s and 30s.
Why? Why would the healthiest people die the most?
The leading retrospective theory is that their immune systems were actually too robust. When they encountered this totally novel, highly aggressive virus, their young immune systems reacted with devastating, unchecked violence.
That cytokine storm we talked about earlier.
Yes. The cytokine storm was so severe that their own hyperactive immune response essentially dissolved their lung tissue and drowned them in inflammatory fluid within hours. The stronger your immune system was, the deadlier the reaction became.
I want to actually jump ahead to our outro concept a little early here because it perfectly fits the 1918 story. The outline specifically mentions the modern reconstruction of the 1918 virus. And this wasn't just historians looking at old medical charts. This was some real-life Jurassic Park-level science.
It really was. In the late 1990s, an incredibly determined pathologist named Johann Hultman traveled to a tiny remote village called Brevig Mission in Alaska. Back in 1918, the flu had wiped out almost that entire village in less than a week. The victims were buried together in a mass grave dug into the permafrost.
So they were permanently frozen.
Frozen perfectly in time for 80 years. Holden obtained permission from the village elders to carefully exhume the mass grave. He managed to locate the remarkably well-preserved lung tissue of an obese woman he respectfully called Lucy. And from that safely frozen tissue, utilizing advanced PCR techniques, scientists at the Armed Forces Institute of Pathology were able to extract the fragmented, degraded RNA of the original 1918 virus.
And they actually rebuilt the code.
They painstakingly sequenced the entire viral genome, every single segment. And that genetic ghost rising from the permafrost told virologists something incredibly shocking.
What did they find?
Well, we'd always assumed that 1918 was a complex reassortant virus, a messy genetic mix of human and pig and bird segments, just like the pandemics of 1957 to 2009.
The pig-mixing vessel theory.
Right. But when they looked at the completed 1918 genome, the genes looked almost entirely avian. It looked incredibly similar to a pure bird virus that had somehow acquired just a few key point mutations that allowed it to adapt directly to infecting humans.
Wait, so no pig blunder was involved?
No intermediate pig required. The virus learned to pick the human cellular lock entirely on its own through pure mutation. That is a deeply chilling discovery because it fundamentally means we can't just blindly watch the global swine populations for the next reassortment event.
Right.
The next catastrophic big one could theoretically come straight from a wild duck directly into a human.
That is genuinely unsettling, and it brings us directly to the modern looming threat the text highlights. H5N1, highly pathogenic avian influenza, bird flu. We've been hearing terrifying news blurbs about it ever since the Hong Kong outbreak in 1997.
H5N1 is the ultimate sword of Damocles hanging over public health. It has been periodically jumping directly from infected poultry into humans for nearly 3 decades, and the kill rate is completely terrifying. In laboratory-confirmed human cases, the mortality rate is consistently over 50%.
That is basically Ebola-level lethality, but airborne.
It is. But, and here is the only biological reason we are still sitting here safely discussing this today, it currently does not spread easily from human to human. Almost every single human victim caught it directly from handling a sick bird.
Why, if it multiplies and kills us so efficiently, why can't I sneeze it onto you?
It comes all the way back to those sialic acid receptors we talked about at the very beginning. Remember the different shapes. Humans have the alpha-2,6 receptors in our upper airway, the nose and throat.
Right.
Birds have the alpha-2,3 receptors.
Right.
Well, humans actually also have those bird-type alpha-2,3 receptors, but they are located exclusively deep, deep down in the bottommost tissue of our lungs.
Oh, so the bird flu physically bypasses the nose and infects deep in the lungs, causing that massive fatal pneumonia, but it can't replicate in the upper airway.
Exactly. And because it's not actively replicating in your nose or throat, you simply don't cough or sneeze massive viral loads of it out into the air to infect the next person. It gets trapped deep inside you.
Viruses mutate.
That is the absolute nightmare scenario. The fear that keeps virologists up at night is that H5N1 naturally acquires a specific point mutation in its HA spike that suddenly allows it to firmly bind to the alpha-2-6 receptors in the human nose while completely retaining its 50% lethality, if that genetic shift or drift happens.
We are in completely uncharted, disastrous territory.
We truly are.
Okay, let's inject a little bit of hope here. Section 6. The defense. We aren't entirely helpless against this shapeshifter. What medical weapons do we actually have?
We have two main pillars of defense: antivirals for treatment and vaccines for prevention. Let's start with the antiviral drugs. Historically, the first major weapons we deployed were amantadine and rimantadine.
Those were the M2 blockers, right?
Spot on. They were designed to act like a chemical cork. They perfectly plugged up that M2 ion channel tunnel on the virus surface. No acid flows in, the internal core never uncoats, and the replication cycle completely halts. It was a simple, highly elegant mechanism.
I hear a but coming.
But, as we've learned, the flu virus is a master of rapid mutation. Over the decades, almost all currently circulating strains of influenza simply mutated the physical shape of their M2 tunnel just enough that the drug molecule no longer fits inside.
They just changed the locks.
They changed the locks. And remember, influenza type B never even possessed that specific type of M2 channel in the first place, so the drugs were always useless against B. Today, amantadine and rimantadine are considered almost entirely obsolete.
So what do doctors prescribe now? We use the scissors blockers.
Yes, the neuraminidase inhibitors. The most famous one is oseltamivir, which everyone knows under the brand name Tamiflu. There's also an inhaled version called zanamivir.
How exactly do those work chemically?
They physically gum up the cutting mechanism of the scissors. The drug molecule binds tightly to the active catalytic site of the viral neuraminidase enzyme. So the virus successfully infects the cell, it replicates, it bugs out of the membrane, but then it completely fails to cut the sialic acid cord. The new viruses just stay permanently tethered to the dying host cell and eventually degrade.
But there's a significant catch with Tamiflu too, isn't there?
The timing is highly critical. You absolutely must start taking the inhibitor within the first 48 hours of your symptoms appearing.
Why the tight window?
Because viral replication peaks incredibly early. By day 3 or day 4 of the flu, the virus has already finished its massive replication phase, and the damage to your lungs is done. Trying to medically block the viral release at that late stage is basically like slamming the barn door shut 3 days after the horse has already bolted. It won't help you.
So if treatment is that tricky, prevention has to be king. Let's talk about vaccines.
The annual flu shot is our absolute best defense. The traditional shot is an inactivated vaccine. Scientists look at global data, predict the 3 or 4 specific strains most likely to cause trouble this winter, grow massive quantities of those specific live viruses, usually in millions of embryonated chicken eggs, then chemically kill the virus completely, and heavily purify the HA and NA surface proteins to inject into your arm.
I have to stop you there. Why are we still using millions of chicken eggs? That seems so incredibly archaic for modern medicine.
It is incredibly archaic. It's literally 1940s manufacturing technology. But the honest answer is that the virus grows incredibly well in eggs, and the global infrastructure was already built.
But it must be slow.
It is agonizingly slow. It takes many months to formulate and brew a new batch. And the source outline highlights a massive, highly dangerous supply chain vulnerability inherent in this egg-based system.
Let me guess. If the next pandemic is caused by a highly lethal bird flu
that rapidly kills off all the commercial chicken flocks,
then we suddenly have absolutely no eggs available to manufacture the vaccine needed to save the humans.
Exactly. It is a terrifying loop of doom. That exact vulnerability is why there has been a massive, well-funded push for modern cell-based vaccines. Instead of eggs, we now grow the virus in giant stainless steel bioreactor vats filled with cultured mammalian cells. We've been using this cell-based tech since around 2012, and it is much faster, cleaner, and infinitely more scalable in an emergency.
There is also another very different vaccine mentioned, the nasal spray vaccine, the live attenuated one.
This is a truly brilliant bit of genetic engineering. Instead of a dead, chopped-up virus, it uses a live, intact virus. But it has been highly cold-adapted.
What does cold-adapted actually mean in the body?
Scientists genetically force the virus to mutate so that it can now only efficiently replicate at around 25 degrees Celsius.
Which is the temperature of the relatively cool inside of your nose.
Exactly. But it physically cannot replicate at 37 degrees Celsius, which is the warm core temperature of your deep lungs.
Well, that's clever.
So when the doctor sprays it up your nose, it actively infects your superficial nasal lining. Your immune system sees a very real, live viral infection happening and mounts a massive, broad mucosal defense.
But it's safe.
Totally safe. Because if those live vaccine viruses try to travel down your windpipe in your lungs to cause pneumonia, they hit that 37-degree heat wall and their enzymes completely freeze up and fail. It's a beautifully designed safety mechanism.
That is incredibly clever engineering. Finally, I want to touch on the absolute lowest tech defense of all. The one our moms constantly taught us.
Good old-fashioned hand hygiene.
Does it actually work against a microscopic machine this highly sophisticated?
It works entirely because of the virus's physical structure. Think back to the very beginning of our conversation. Remember that stolen lipid envelope, that greasy coat of fat holding the whole thing together?
Bloody jacket.
What exactly does common dish soap do to grease in a frying pan?
It dissolves it.
It violently dissolves it. When you actively wash your hands with warm water and basic soap, you are chemically dissolving the virus's protective skin. You completely shin away the lipid envelope, and all those perfectly evolved HA and NA spikes just wash away down the drain. The entire viral matrix literally falls apart into harmless protein junk. Alcohol-based sanitizers do the exact same chemical disruption.
That is weirdly empowering. I can completely destroy this pandemic-causing death machine with a 50-cent bar of dial soap.
The basic laws of chemistry and physics are entirely on our side there, yes.
Well, we have covered a truly massive amount of biological ground today. We've gone from the deceptive, slow drift of the seasonal flu that ruins our winters to the catastrophic, sudden shift of the pandemic flu that alters history. We've looked deep at the segmented genome, that 8-card deck, that makes this virus such an unparalleled master of disguise.
We've explored the intricate cap-snatching nuclear heist and the brutal mechanical efficiency of the spring-loaded HA grappling hooks and the NA escape scissors.
And we've looked at the sobering history. The genetic ghost of the 1918 virus rising from the Alaskan permafrost to starkly warn us that nature doesn't always follow our neat little textbook rules about pigs and mixing vessels.
It's a very humbling reminder that influenza is not a solved scientific problem. It is a highly dynamic, constantly evolving biological force that is always probing our defenses.
So what is the final practical takeaway for the listener today?
Simply put, respect the flu. Do not underestimate it as just a bad cold. Go out and get your annual flu shot. Because even if the strain prediction isn't perfectly matched that year, the partial immunity is often exactly what keeps you out of the ICU and off a ventilator. And for the love of science, wash your hands thoroughly.
And I suppose if you happen to be on a farm and you see a sick pig and a wild duck hanging out closely together, maybe just run the other way.
That is exceptionally sound epidemiological advice.
Thanks for taking this incredible deep dive with us today. Stay healthy, everyone.